Welcome, everyone, to our version of the Texas Rodeo. We have great speakers, Kenny and Ryan. I'm going to turn it over to them, but before I do, I just want to thank them. They've done a spectacular job organizing this workshop, and from this point on, they're in charge. I'll go mute. So again, thank you, Kenny, Ryan, and thanks to all the speakers for joining us from across the world. So thank you.
Thank you, Stan. Once again, I want to welcome everybody to this program. We are so glad to have you, our speakers, our attendees. Thank you very much. Myself, Kainde, my name is Doru Kainde. I'm a molecular and a virologist and a computational biologist, a research scientist at Texas University. And together this afternoon, my co-host will be Ryan. Ryan, can you please introduce yourself?
Hi, everybody. So my name is Ryan Gutimi. I am a medicinal chemist postdoc here at Baylor College of Medicine in Houston in Gene Wong's lab. And yeah, I'm working mainly on developing small molecules targeting the soluble epoxy hydrolase for Alzheimer's treatment and also Alzheimer's disease treatment, and also with a separate funded fellowship working on developing selective PKMYT1 inhibitor for cancer treatment. And yeah, welcome everybody. And we are all glad to have you here and hope you'll enjoy this workshop and take some information from it. Thank you.
Thank you, Ryan. Thank you very much. So our first speaker for this afternoon is going to be Krishna Govender. Krishna is from the University of Johannesburg, South Africa. So Krishna, can you please start your presentation? 10 minutes.
Okay. Give me a second. Just let me know when you can see it.
Yeah, we can see your slide.
Okay, so guys, I apologize in advance. I didn't know my crowd, so things might be a bit basic, but hopefully I can give you all some input as to what advancements we've made in things like molecular docking and dynamics over the years, right? So why do we need this thing called molecular modeling? So essentially, we tired of doing things the old way, where you work in the lab and you find like 10,000 ways that don't work, right? So we currently now have steered resolutions to doing stuff. So we know where the target is, what we want to do with it, and we want to find a drug which we can design via computational methods to try and find some solution, right? So for those of you that don't know, which I highly doubt, so if you think about what is a drug, so it's just a small molecule that binds to the target, such as a protein or enzyme, and it has certain functions that can turn on or off specific biological or psychological or physiological processes in the body. Right, so now we can imagine the drug as being a key to our lock, which is in our enzyme or protein. Now we need to find from this bunch of keys, the key that fits into this protein, right? That will bind the best and work on the target. So of course, how do we do this? We can do this experimentally. So experimentally, we use high throughput screening, which a lot of you would probably already know. But this process can be quite expensive because you now need all of these drugs in here before you can actually administer them, right? So it's costly. I apologize. I understand you're in the US. Those are ants. So the alternative is using virtual screening, right? Where we then use computational models to actually now try and screen as many drugs as we can to try and find targets that will actually work. So let's imagine this is our drug landscape. So the biggest thing is that if you do this through experimental methods, you maybe isolate drugs that sit within, let me quickly just put my laser pointer on, that probably maybe sit within this region. But what if the drug sits somewhere here? The likelihood of you sampling that is going to be quite low, right? So if you use computational methods, we can actually sample a lot more of this drug landscape because we can dock many more ligand compounds or drug compounds. This is just a list of different drug databases that we currently have available to us, right? So you guys can go visit these. I think I give the links on the side there. Maybe I should minimize my thing so I can see. Yeah, so I do give the links there. And you're able to actually download these ligands and then use them as possible drug candidates if you wish. Right, so now, where does this whole thing of computer-aided drug design come in? So this concept of computer-aided drug design gives you one of two approaches, which is a structure-based or a ligand-based drug design, right? So from your structure-based drug design, you may know your protein structure, you have a fully resolved crystal structure, so you can then dock onto an active site. Alternatively, you don't know the structure, so you have to use something called homology modeling, where you can then predict the pieces that are missing of your protein and then do your docking. If you can't do homology modeling, then you can go to the ligand-based drug design approach, right? Where if you know your actives, which are active ligands, you can then either do a similarity search, pharmacophore mapping, or QSAR models. Alternatively, if you don't know if you know your actives and inactives, then I see a lot of people here will probably be speaking about machine learning today, so you'll get to learn a bit more about that as well. Right, so this is just a basic structure of how you would conduct something like molecular docking. So I'm not going to walk through all of this due to time. But what I do want to show you here is that you have many different sort of docking applications, whether it be standalone applications that you can run on a high-performance computer or even on your standalone machine, right? And then you have some that are online applications, which you then submit your molecules online, and then you can do the docking via those routes. So here, there's just a few theoretical values that I've thrown here, right? So say you have 2 million of these drugs that you wish to dock onto a certain protein, and you do it on a single core processor. It can take you up to 46 days just to do this, right? If you're docking 2 ligands per second. Now, as technology has advanced, we've now reached a point where you can actually do docking on graphical processing units instead of just playing games on those things, right? So now you can actually do much more in a shorter period of time. So the exact same thing which would have taken you 46 days initially can now go up to 13 minutes to dock this many ligands. Right, so the other approach we use is molecular docking, dynamics. So for dynamics, what we do is you take the docked ligand, which is sitting in your active site, and then you run live dynamics on it, where you actually can see the motion of the molecule during a certain period of time. Now, often we do nanosecond range because that's what we actually can get up to with the current technologies. And you'll notice that in some cases, you want the ligand to bind to the active site. Other cases, maybe the protein is just a transport medium. So it transports the ligand somewhere, and then it just throws the drug out to go do its job somewhere else, right? So here I give you a list of just a few molecular dynamics packages that I'm familiar with, and there are probably more, but there's also, so again, there's academic versions and then there's commercial, right? So you guys can go and visit the links that are here. And here again, I just show you, look, if you look at the advancement in technology over the years, right, compared to, so what you want to do is you want to get as many nanoseconds out of some simulations as possible. Now, compared to the conventional technologies, which were back in the days, which is just normal CPU processors, where we used to get something like 0.29 nanoseconds per day for a system like this, when you look at the current graphical processing units, you can actually go up to things like 39 nanoseconds per day for exactly the same simulation with the same solution. So technology is advancing, and this is where everything seems to be headed, is this GPU technology. Of course, the physicists are currently busy with quantum computing, which is the next step, but that's a story for another day. And what I'm also looking into is QMMM, where we try to combine quantum mechanics with the molecular mechanics so that you can actually sort of view reactions inside the active site of enzymes, because we know that those are actually things that speed up reactions, right? So yes, from my side, that was basically all I wanted to tell you today. So I just like to thank the organizers for inviting me to give you all this talk, and thank you for listening.
Thank you, Krishna.
Thank you, Dr. Govender. Now we will turn it to Dr. May. If you can share your screen from the university, okay, from the Wisconsin Institute of Discovery.
Thank you. Thank you for the invitation. Let me share my screen. Right. And I will do my best to be timely, and thank you for the organizers for inviting me, particularly Kenny. Thank you for reaching out. And Kudimi. Okay, so today I'm going to try to quickly talk about how we're developing models for host pathogen interactions, and I'll try to point out opportunities for where I see AI playing a role and being rather important. And also I'm going to tell you a little bit about where we looked at small molecule interactions with our systems biology models. And so our work, we're interested in infectious diseases, many different types, specifically Mycobacterium tuberculosis, which many of you are probably familiar with, and the fact that it's one of the top 10 causes of death worldwide. It's an important infection. It's a lung infection, and there's many different things that are hallmarks of TB infection, specifically the formation of this granuloma. And the granuloma structure is something that many people have been studying both experimentally and computationally and building mathematical models around it, because in understanding this granuloma structure, we can understand something called latent TB infection, which tends to be probably about 25% of the population is walking around with latent TB infection, and which is a challenge, particularly when people become immune compromised. So the structure is actually part of the way that TB is able to survive inside the host. So one of the goals is trying to understand how can we target TB when it's in the structure, and also, what happens when the structure breaks apart. And even though the structure can be protective both for the host and also for the pathogen. And so there's many different models around it. There's different models of the pathogen where you're trying to understand the bacteria and how it survives even as it's in an oxidatively stressed environment. And so our approach to looking at it many times, we're very interested in the dynamics of the interaction, the dynamics of how the TB is surviving and metabolically adapting. And that's kind of what I'm showing here on this x-axis, as well as the dynamics of how the host is changing the environment that the TB sees, which I'm showing on the x-axis. And so our interest is how the interaction of those two dynamics leads to basically the outcomes that we see related to the disease. So I'm going to tell you a couple of quick stories. One is some of the models that we build are either pathogen focused, some are host focused, but really our excitement is when we can integrate the two. So when we look at TB, one thing that's very important, as you can probably guess, it's in the lungs. So oxygen is extremely important. And so there are some experimental models of TB and how it responds to oxygen. One very famous one is the Wayne non-replicating persistent model. And that's kind of what I'm showing with this cartoon here. And so under that condition, you look at what they observed is that under, of course, well-oxygenated conditions, TB is very happy, it survives. But when you slowly remove oxygen, which is shown in this middle graph by slowly stirring it and growing it, TB is still able to survive even in the absence of oxygen. But when you rapidly remove oxygen, TB dies. And so we've taken that experimental model and the data from there and tried to develop a systems biology model that looks at the TCA cycle, as well as the electron transport chain within TB. And we build mathematical models around it to try to investigate how TB actually survives under these different conditions or dies. One of the key pathways is this glyoxylate to glycine shunt that has been observed in TB to be upregulated, where the bug upregulates glyoxylate and actually uses it to produce energy molecules through the ETC. So we've taken a lot of data, and part of the reason why I'm showing you this graph is just to show you all the different pieces of data that we integrate into our systems biology model. So it's beyond writing the equations, but we're pulling in information from experimental papers such as ATP levels, genomic data, microarray data, in the old days transcriptomic data. Now we're using that to build regression statistical models that we then integrate into our systems biology models to try to predict outcome. And so we've put together models like this. These are our ODEs, mechanistic-based models, which differ from AI-based models. But these mechanistic models are really a bottom-up approach to try to understand the system. And so one of the outcomes of our models is shown here, where we can predict the ATP levels. We compare it to what's going on. We compare it to what the data says. And so on the left, you see kind of the levels of ATP over time and how they match up. But the real interesting thing here is that we can use this to then ask the question during these different stages of adaptation of the bug to different environments, what's happening. So we can, and what we've seen is that when TB tends to die, what it's doing, it doesn't upregulate the right pathway. So on the top, you see here that the TB is actually downregulating specific enzymes early on versus when it survives is upregulating various enzymes early on. And so we've used these types of systems biology models to ask questions about what the pathogen is doing in order to survive. And the other thing that we've done is we've then integrated these models into multi-scale models. Sorry, I don't know what's going on with my pointer. We've integrated this into multi-scale models. Let me go back here. There we go. Just show you this one real quick. Sorry, my computer's acting. Okay, so these multi-scale models, they're taking the mechanistic model and they're combining it with other types of cellular models. And then from this, what we're trying to do is learn and understand how the structure interacts and what the bug is doing inside the structure. So this is an example of TB and the ATP levels inside of the bug. And so as you can imagine, these types of platforms are very ripe for opportunities for integrating things like what happens when you introduce a small molecule into the system. And with these types of models, we're able to replicate what's measured experimentally inside animal models of TB, and that's what these comparisons are showing, like in terms of the number of granulomas and the number of CFUs that are found in the granulomas. So basically, the number of bacteria that are found in the granulomas, we're able to replicate it with these types of models. And so, as we build these models, what we really want to do, and what some other people have already shown, is then take the models that we built as a platform for exploring what happens when you introduce different small molecules in terms of drugs. And so some early work that the collaborators and I worked on is this field called systems chemical biology, where basically we use small molecule information and the interaction between small molecules and enzymes and introduce that into our systems biology models to see what happens. And so the previous talk was great because it actually talks to you about docking, which is not my expertise, but our collaborators, they basically did a lot of the docking to explore different types of inhibitors for specific enzymes involved in this TB pathway. And they use various tools that the previous speakers discussed. But what they do is then they give us these small molecules that they've already used their computational drug design platform to say these are possible interactors with the enzymes of interest. And we include that in our simulation. And what I'm showing you here is the second graph here is simulating, it's a malate synthase inhibitor in the TB model where there's well aerated system versus the slide B, the graph B is showing you under low oxygen conditions. And the important thing here is that you see that you actually, at least the simulated response shows two different types of responses depending on the environment that TB finds itself in. So as you can imagine, this is kind of a platform that allows us to not only simulate and understand how the bug is interacting with the host, but then when we introduce small molecules, we might see different actions or mechanisms of action depending on the condition that the pathogen is sitting inside of. So in summary, I don't know if I've gone over, and I apologize if I have, but the point here is that we do have this platform. Our mechanistic modeling is able to help us integrate different types of models and different types of data sets. We're able to see physiologically relevant outcomes. But one of the challenges is parameter space, right? So mechanistic models are very trying to find the correct parameters that match the data that you have is a challenge. And I think this is where we've actually used quite a bit of AI types of platforms, very generic ones such as, we're doing like genetic algorithms or things like that, but there's much more advanced AIs that can help us. The other thing that we've also found is that sometimes we don't have a single mechanistic model to fit every environment. And this is where we can come up with basically sets of models, right, that actually represent our system. So using AIs to help inform that kind of decision or that kind of finding basically sets of suboptimal models, but that are better together to actually represent the outcome is another area that we are very interested in with respect to AI. So I'll stop there and hopefully there'll be questions and opportunities for discussions. So thank you.
Thank you very much. Thank you very much, Dr. May, for your presentation. Thank you very much. Our next speaker is going to be Dr. Pora from Northeastern University. We can't hear you.
That makes more sense. Sorry, I muted myself.
Yeah, so
Thank you for not letting me go for 5 minutes. So thank the organizers for this opportunity to speak, and I look forward to hearing about from the other speakers as well. This is a great group that you pulled together. So I'm going to talk about a different aspect of this problem, whereas Krishna was talking about finding the keys to the locks. Our goal is to find new locks for which you need keys. So we're using simulation methods to look at systems where we don't really know how we could stop them or target them. And maybe we can find new targets through our methods and then hand it off to other people who are really the experts in targeting these systems. So I'll just give 2 recent examples of systems we've looked at where we have some implications for potential strategies for targeting. And the first one is the ribosome, which is an antibiotic target. Ribosomes are in every cell. They're unique to different organisms. So the antibiotics work by shutting down the ribosomes in bacteria, not hurting the host, and not hurting the mitochondrial ribosomes in the host as well. So what we do is we look at all of the conformational rearrangements that occur during the elongation cycle of the ribosome, and we try to identify what are the rate-limiting interactions, where are the free energy barriers, can we perturb those free energy barriers and therefore perturb the kinetics. So if we can establish a relationship between structural aspects of the ribosome and dynamics, then we can say, well, this is a way you could target the system in principle. So in a recent study, we were looking at a motion called hybrid state formation, which is basically just where a tRNA molecule moves between one site called P to the other site called E. For this, we simulated hundreds of transitions between the two sites, and through a statistical analysis of the trajectories, we identified 6 residues that seemed to be most important in controlling the kinetics. We then went back and actually perturbed those residues. It just artificially made them a little bit larger, just to say, if we were to put a small molecule here, would we expect there to be a big effect on kinetics or not? And so we did that, and what we found is that there are few of the 6 residues are actually more important than others. And in particular, there's this protein called L33, where there's a loop, ASN29 loop in bacteria. It's leucine in human mitochondrial ribosomes, and it's a different protein altogether in human ribosomes. And if you look across all bacteria, this loop is actually quite very well conserved across bacteria. So again, we're not a drug design group, but this would suggest if you can put a small molecule here and just get it to stick in this region, it should actually have a very big effect on the kinetics of bacterial elongation and ideally not affect the host at all. So we propose this could be a possible target for broad spectrum antibiotics, but hope maybe someone here will see this and think of a great way to test that idea. We're not drug design experts ourselves, but we're very excited with this possibility. Another example that a lot of people have been looking at is obviously the SARS-CoV-2 viral capsid and the spike protein. So everybody's probably seen this image before, where you have the spike proteins flanking the viral capsid. And from our goal is, can we learn something about how the spike protein works? Maybe there are transient interactions that are formed, or transient regions that are exposed as the spike protein functions, and those could be targeted to potentially develop better antibiotics or vaccines or antivirals. So for those of you who haven't looked at the spike protein carefully, this is sort of the process of what it does. There's the full spike protein. When it recognizes the host membrane, the S1 subunit is recognized and then dissociates, leaving the S2 subunit. And at this point, there's a certain amount of time that the S2 subunit can extend the fusion peptides, these long tails, try to attach to the host. And if it does successfully, then it can bring the 2 membranes together. And this allows membrane fusion to occur, and then the genetic message can be passed to the host. So this is a mechanism of infection for the virus. There's another pathway as well, but this is one of them. So we wanted to ask, can we learn something about this rearrangement? All we know from experiments are the endpoints. So the middle picture and the picture on the right are what we know. And the protein essentially inverts itself, goes from the inside goes out and the outside goes in during this process. It's a pretty wild conformational motion. So can we identify something that is potentially distinct about this rearrangement that could be used to target it? So I'm not showing any of the data for any of these, either of these projects for a 10-minute talk. So I'll just show an overview movie of what one of these trajectories looks like, and I'll tell you what the results were. I run the risk of people thinking that we drew any conclusions from the movies themselves, which we don't. This is one of 1,000 simulations. So all of our claims are based on statistics. So this is how the simulation starts. You have the S1 or S2 subunit. S1 is dissociated, and we bind it, we mimic a membrane, and then we just have to let it undergo its conformational rearrangement to this post-conformation. And what we see here is actually this head domain in red gets stuck along the way, which we attribute to the glycans actually inducing this pause. And the idea is that this pause actually gives the fusion peptides more time to try to successfully reach the host. So this pause is consistent across all of the calculations that we performed. It goes away when you get rid of the glycans, actually. And here you have the fusion peptides basically searching. And if they're given enough time, they will eventually be able to bind to the host. In this particular simulation, the viral membrane was included. The host was not. The host is just illustrated here, but we did include it in other calculations, and the overall result was the same. And so after it successfully binds the host, this conformational rearrangement continues, and it sort of zippers up, and that's where the energy comes to bring these two membranes together. And so in this process, this big blue helical region was exposed for a very long amount of time. It's not exposed the same way in the pre or post configurations, but it has to sit there for quite a while during the fusion process. So that's another target that we would suggest may be useful for vaccine development or antiviral development as well. So those are just two examples from our recent studies of how we're trying to provide insights that can be useful in drug design studies as the next step. That's all. I'll say thank you to everyone to the work. So thank you.
Thank you, Dr. Whitford. Now we will turn it to Dr. Frank, if you can share your screen from the Arrakis Therapeutics.
Okay, so thanks to the organizers for including me in this session. So my name is Aaron Frank. I'm currently the director and a head of computational chemistry at Arrakis Therapeutics. Prior to that, I was an assistant professor at the University of Michigan, running a computational biophysics group centered around modeling RNA structure and dynamics. So what I'm going to do here today is talk a little bit about some of the work that I did as an assistant professor in terms of building machine learning-based tools for interrogating RNA structure. So why RNA? RNA, just like proteins, regulate a lot of important functions in the cell. And there is no growing interest in RNA as a drug target. So currently, there are a few examples of small molecules that, if you look at them, they look very much like small molecules that would bind to proteins, but they actually target RNA. The interesting thing about the molecules that I'm highlighting here is the fact that these were all identified via phenotypic screens. So that means that it's only after the fact that they were we realized that they actually target RNA. So that means that the discovery of these molecules was not intentional, right? The fact that they targeted RNA was not an intentional process. So at Arrakis, the approach is one where you start with RNA structure and then you use screening, high throughput screening to identify potential binders. And obviously you follow that up with functional and mechanistic assays. So I'm a computational biophysicist, and so my focus is always on RNA structure, right? So the task of trying to identify small molecules that bind to RNA starts with structure. So the basic process is this, and Krishna kind of highlighted this already, so I don't need to spend a lot of time here. And you start with a structure, you do some mapping of a cavity to identify a region where the small molecule is likely to interact. And then the goal is to then find small molecules that fit neatly into these cavities, right? And so here you're using docking to do this. So central to this is understanding the nature of the interactions between RNA and small molecules. So looking at RNA ligand complexes is something that I spent a lot of time working on. So the number of aspects of this that are important. One is structure. So thinking about the structure of the RNA, which is the target overall, but also thinking about where on that RNA the ligands are likely to bind. Once you do that, you also need to understand the RNA ligand interactions. So thinking about RNA ligand poses. Energetics is also important, right? So understanding ligand binding energies, and kinetics is the last thing, right? Where you want to also understand ligand unbinding rates and also the pathways involved. So what I'll do today is just give you a couple of examples of how we use machine learning to solve some of the modeling tasks associated with the first two of these, which is really about mapping cavities and also looking at RNA ligand poses. So one of the tools we've developed, we call the RNA cavity miner, right? So essentially what we did here is to train machine learning classifiers that can recognize what a ligandable pocket is. So essentially you look at a RNA, you examine the surface of the RNA, and based on some of the properties of the surface, you can actually predict whether or not a small molecule is likely to bind here. So we use machine learning to be able to do this. And the basic pipeline is one where we start with known ligandable or druggable cavities, right? We place what we call these pseudo-atoms at particular sites, and then calculate a bunch of features associated with those pseudo-atoms, I should say. And the idea is that the features projected onto these pseudo-atoms describe the cavity. And so if you build such a database of ligandable pockets and also non-ligandable pockets, you can actually learn how to distinguish these from their features. And so I'm not going to go through all the details. So we had about 88 RNA ligand complexes, and we were able to use that data to train these classifiers. And the performance is reasonable, right? So we're able to recover actual ligand binding sites with a AUC about 0.93. Just for reference, if you have a really good, a perfect classified AUC is about 1, right? Something that's about random is about 0.5. So here we're seeing values close to 0.9, which means that they're doing a decent job of identifying the sites at which ligands are likely to bind. So the next stage is, okay, you've identified a site where a ligand is likely to bind, you're going to do some docking. But one of the known challenges in docking is the fact that very often you can sample many different poses, and it's very hard to choose the correct pose, right? Even if you have a scoring function that you use to guide your docking, very often the most favorable pose as judged by that scoring function does not lead to the best or the correct pose. So here again, we use machine learning to kind of help us address this task, right? So you would do your docking and have a collection of poses, and then run those poses through a classifier. And here again, these classifiers were machine learning based. So I'm just going to show you an example of this application. So what you're seeing here is an example of the actual pose and the pose that's predicted using the classifier. On the top, I have a couple more examples. The first two are cases where we failed, but the others show that we're actually able to disambiguate the native pose from a set of decoys. Now, the workhorse of this approach is being able to fingerprint the RNA or the region around the RNA. And so we have atomic fingerprints that we can use, and that's useful for when we were building the RNA cavity miner. But also there are ways to turn that into a molecular fingerprint such that if you're looking at a small molecule, you can actually aggregate all of that atomic fingerprints into a molecular description of your small molecule. So as the pose changes, this molecular fingerprint changes, and that's actually what we use to be able to predict or classify poses as native or non-native. Now, this kind of approach is really dependent on access to lots of data, right? If you think about AlphaFold, for example, there's a lot of excitement about AlphaFold in the context of RNA protein structure prediction, and the success is really dependent on the fact or reliant on the fact that you have tons of data that you can use to train these models, right? So over the years, thousands of crystal structures have been deposited, and we have the data that we can train models on. Now, for RNA, one of the major roadblocks is the fact that we don't have a lot of structural data, right? So what I'm showing you here is the number of solved structures in a PDB for proteins, that's shown in blue. And by comparison, you see what we have for RNA, right? So it's really, really tiny. So this is what I think is going to be a major roadblock to progress as it relates to applying some of the exciting machine learning approaches for proteins to RNA. So if you don't have data, there are instances in which you can create the data, right? And so one example, one possible way to do this is to generate data from physics-based simulations and then learn from that data, right? So what I'm showing you here is an example where what we're going to do is to use simulations to generate unbinding trajectories for a given collection of poses. So you have a set of poses here in different colors corresponding to different poses. And for each of them, you can generate unbinding trajectories, and these paths here just show how these individual poses leave the pocket. And then you can do contact analysis and come up with an unbinding profile for each of these, and you can fit them to some equation here. And what you can do is then learn how to essentially recapitulate this experimental or the simulation derived fit directly from the initial pose, such that you are able to now compute these kinetic parameters directly from structure without having to do these long simulations. So the power here is that the fact that you can actually generate this data yourself. And so in cases where you're lacking data, you can actually use these kinds of physics-based simulations to generate the data. And so we applied it to the post prediction problem. And so what I'm showing you here on the top are examples where we have a bunch of different poses shown in pink, and we try to resolve the native-like pose, which is in green. And the way we do this is to run them through our classifier that was trained on the simulation data, right? And actually not a classifier, a regressor that's actually able to predict the kinetic parameters that one would expect to observe if they actually ran the simulations. And based on those predicted kinetic parameters, you can actually sort the ligands, and you can see, for the most part, we're actually able to get the correct pose from this collection. So this is a really neat example of how you can, run simulations to generate the data you need, and then use that to actually build machine learning models. And so with that, I'll say thank you, and I'm look forward to answering questions at the end.
Thank you very much, Dr. Frank. Thank you so much for that wonderful presentation. So without further delay, let's go to our next speaker, and that's going to be Dr. Pora Kim. Dr. Kim, your presentation, please. Thanks.
Yeah, hello everyone. Thank you for inviting me in this good consortium meeting. Yeah, so today, let me, so my name is Pora Kim. I'm assistant professor in School of Biomedical Informatics in the University of Texas Health Science Center at Houston. So today my talk title is Bioinformatics Studies Using AI Approaches for Developing the Targeted Therapeutics in Fusion Genes. Yeah, so since my PhD at the time, from the time until now, I really like this topic, the fusion genes, because I see there are many interesting study topics there. Still, so I think so today maybe it seems like I'm the only person that the background is bioinformatician, but I studied my bachelor was chemistry, so I will try my best. Anyway, so let me introduce about the fusion genes. Actually, you may know, like in the cancer cell, there are many different types of mutations. So based on the length of the mutation, we make we have different types of mutation like this. The smallest one is single nucleotide variants, and indel or copy number variants, or structural variants. So structural variants is the largest DNA mutation often happen in the cancer cells. So you can think like the fusion gene is the byproduct of the structural variants. So the breakpoints of the structural variants, if they are located in the gene body, then we call those as the fusion genes. So in this DNA, and then two different loci, if they're at the same time, this kind of breakage happened, then cells try to repair, but if it can be failed. due to some fact, and then it will make this kind of a rearrange the chromosome structure. And this is the fusion gene. Two genes are fused together, and following the central dogma of biology, it can make a fusion transcript, which is mRNA, and then it will be translated and form the fusion protein. So the best well-known case is the glyvac target, which is a BCL-ABL1. So is I'm also trying to find the kind of good candidate. So these are the mechanisms of action of fusion genes. Like the representative ones are like kinase fusion genes, which is the signal transduction without control. So it will make tumor cells, or target gene activation by the transcription factor fusion gene binding, or it can lose its some important PPI interactions, or can avoid the microRNA regulation. It will increase the kinase activity, or it can also affect to the downstream vectors, something like that. So last year, we made this database, fusion gene database, second version. So this is the most comprehensive database of the human fusion genes. And this time we put some annotation relating genomic features using our developed AI approach, fusion AI. And we also made another model to predict the potential coding of the fusion genes. And then we also have another visualization tool of the functional features of fusion genes. And we also provide the fusion sequence. So this is the only source providing the comprehensive fusion amino acid sequence. So UniProt, they are working to import our data into UniProt. So so far we have more than 100,000 fusion genes, and out of these, there are 16,000 different fusion genes. So for these, we checked the ORF, and then potentially we have now like 43,000 fusion proteins. So you can think like this fusion gene is kind of new genes in human. So this slide shows the fusion AI approach. So using this one, we can predict the input is the genomic sequence, like from the 2 breakpoints, plus or minus 5 kb, then the total size of 20 kb. We just give the DNA sequence, then this tool will let us know whether it will have chance to be used as the fusion breakage or not. So I think this from this model, we could successfully identify some related features. And these days we are working on using those features. We are working on searching some microhomologies relating some DNA repair pathway. So this is the protocol. And we also enhanced the training data set using the real riboseq data, and then we could build a successful model of to infer the potential coding of the fusion mRNA. And recently we are working fusion PDB. This is a fusion protein database. As I explained from our fusion gene database, we have like 33,000 fusion protein sequences. So as you know, 2 years ago, like from the Google team, they made AlphaFold 2, and the University of Washington, they reported a Rosetta Fold. So from that work, we thought we can predict their structures, because the situation is like, even if we visit the PDB, there are not many fusion structures available, very few. And even the title looks like a fusion gene, but many of them are just focused on the specific domain in one gene. So it's not actually a fusion gene. So we can regard like we don't have a whole structure of the fusion proteins. So because so far there's no information of the full length fusion transcript sequence, no resource of the full length of the fusion amino acid sequence, then naturally we don't have any structure about this. Then we can not predict their function. So I wanted to contribute in this way. So if you visit fusion protein database for these 33,000 fusion proteins, we provide the basic annotation plus sequence. And for the level 2, for the 1,200 manually curated fusions and 2,300 recurrent, which have at least more than two presented samples. For those fusions, we predicted the whole structures. And the focus on manually curated ones, we performed the virtual screening across the approved drugs. So these are the number of translated fusion genes. And then when we check the number of published articles, we can see that almost like 98% of them were never reported in study. So we have to go further. So we have to study a lot. So this is kind of after we make a fusion amino acid, then usually we check the domain retention, and this is another refugee viewer we made for each gene, each mRNA, and each protein. We provide what kind of functional features can be lost due to the breakpoints. These are the breakpoints. Inside is we can think like a deletion event. So so far we predicted the structures of these number. So this is the distribution of PLDT score, and then when we predicted the two partner genes, partner proteins structure with AlphaFold2, their PLDT score distribution looked like this. And then when we check the PLDT distribution of a fused one, it seems like the combined one between two wild types. And our assumption was if we can have the whole 3D structures, then based on that, we can predict better active sites because there will be some 3D interactions. That was our assumption. So we predicted those whole structure, and then we compared the docking score. So the first number one is the active sites from our model. The second one is just known active sites. So compared to known active sites, we can see that our active sites have a strong binding with their drugs for BCI-BL1, TMPRSS2. And recently there were FDA approved notice for this kind of NTRK fusions, NTRK1 inhibitor. So when we screened all kinase inhibitors, then the specific NTRK inhibitors have the strong binding when we compare it with our predicted active sites. Yeah, so since my master and PhD, every year I'm trying to study many different aspects of the fusion genes and proteins in diverse way. And this year, we are working on fusion neoantigen. So you may know many different types of mutation can make neoantigens. So fusion protein also will be shredded, and then the breakpoint peptide can be the target can make neoantigen. So since we have all the potential fusion protein sequences, so we are doing this kind of analysis. So we hope to provide some novel therapeutic targets. Yeah. So these are all my presentation. Thank you.
Thank you, Dr. Kim. Now we will turn it to Dr. Antunes from the University of Houston. If you can share your screen.
Okay. See if we can see my screen now.
Yeah.
Okay, thank you for the invitation. I'm very happy to join this team discussing these topics. And I will be talking more about peptide ligands and peptide mimetics. I'm a computational biologist working with structural bioinformatics, mostly applied to immunology problems. And before I go further, I just wanted to highlight that peptides, there is this growing interest of the use of peptides as drugs. There are a growing number of these drugs being approved by the FDA, and peptides have some advantages in relation to small drug-like ligands in terms of bioavailability, in terms of cell permeability, and so on, and also in relation to proteins, like larger proteins as drugs. And these peptides can have different properties like antimicrobial property, cell permeability, the ability to target specific cells or specific proteins, and even sometimes mark these proteins for degradation and so on. So for all these reasons, there is like this growing interest and funding towards the study of peptides and peptidomimetics as drugs. And in this context, I have been doing some structural bioinformatics analysis. I for instance, predicted the binding modes of some peptidomimetic inhibitors for the SH2 domain of STAT3 for cancer research applications. I predicted the binding and the dynamics of cyclic peptide inhibitor for the C3b protein or receptor. And these end up being the first drug approved by the FDA for targeting the complement system, which is part of the innate immunity. But most of my work has been focused on the human leukocyte antigen receptors, or HLAs, which bind peptides from inside the cell and display them on the outside for recognition by the immune system, particularly by T cell lymphocytes. So predicting which peptides bind to HLAs has direct applications for vaccine development and for T cell based immunotherapies. And this is kind of most of my research has been focused on those applications. The gold standard methods for predicting if a peptide binds to an HLA are neural networks, which are trained in available data sets, experimental data sets on binding affinity for these peptide ligands. And they have been used for, vaccine development, for personalized immunotherapy, but they have some limitations. One of them is the lack of interpretability of the predictions, and another is the requirement for large data sets. And one challenge here is that there are so many different HLA alleles, and each one with different specificities. So what happens is that these models are very good for some of the alleles, but not as reliable for other alleles, given these biases on the training data sets. So one thing that I'm trying to address is if we can improve these predictions and related predictions using structural information. So I started using molecular docking tools that is the gold standard for predicting how a ligand binds to a receptor. But the problem here is that as you know, these software are more designed to work with small drug-like ligands, so with up to 10, 15 flexible bonds or degrees of freedom. And these peptides that I'm interested in, they have sometimes over 50 degrees of freedom, so they are too big. And these bring some challenges both for the sampling, or how to explore all the alternative conformations of this ligand, but also for the scoring. So how to actually account for the sheer number of interactions that this small molecule can make, and account for that on predicting binding energy. So I have addressed, I worked on both of these problems, on the sampling more using geometric approach, and on the scoring now using more machine learning. And I'll talk briefly about those. So just to give one example on the sampling side, one software that we developed was this incremental method called DINK, which instead of dealing with the whole ligand at once, it selects a fragment of the ligand, let's say with 6 degrees of freedom. It runs a regular docking tool for finding the binding mode for that fragment, and then it grows this fragment, adding an incremental piece of the original ligand, select a new set of bonds to be flexible, runs the docking again, and keeps doing this incremental process up to the point that we have the whole ligand docked. By combining this incremental process with parallelization, we are able to do these predictions in a more efficient and more accurate manner as compared to regular, the use of regular docking tools. This is a general method we use for peptide HLAs, but can be used for any receptors and ligands, and it has been used for many users in different countries. And we started using these methods to model peptide HLAs, but we also developed some peptide HLA specific tools that are even more efficient. And we start asking this question, can we use these structural models as additional training data to improve the scoring, for instance? So the first attempt we did in that direction was headed by this CS student from Rice University, J.V. Abella, and he selected a data set of peptide HLA complexes for which we have data. It ran structural modeling using one of the HLA-specific methods we developed. So generated a large data set with over 300,000 complexes, and used this complex to extract features, geometric features like distances between amino acids or distance between atoms, and use these features to train a random forest model. In this case, was a classifier, so it was predicting binder, non-binder with some probabilities. And we had some very good results, which both validated the quality of our models and the possibility of extracting these features and using these structural features. But we had another student that pushed this even further, trying to train a model to predict binding affinity. And the traditional way of predicting binding affinity, as you know, is using more kind of biophysics, biochemical-based methods. So here is a representation of a scoring function from Rosetta package that she used as a baseline. And she trained a machine learning, a random forest model for the peptide, based on the peptide HLA structural interactions like I described before. And she also trained another version that was in which she was taking features for each one of the peptide positions. And in this context, it makes sense because some positions are more important than others for the binding to this receptor. So it turns out both these machine learning models work better than the standard scoring function of Rosetta for these complexes. And you can see that we tested across different HLA alleles to show that it was not biased to one specific receptor. The idea being that these structural features are more generalizable than, sequence data sets for a given allele. And the per peptide position scoring function outperformed the standard model with the whole peptide in one set of features. And we also compared the performance of this scoring function against other common docking scoring functions like from Vina, Vinardo, Autodoc. And we did better than these other scoring functions when ranking peptides, peptide structures with this scoring function. The last point I want to make here is talking about one of the main challenges of these machine learning methods is exactly the availability of training data. And in the context of peptide HLAs, we have lots of data on binding affinity, although it's still biased, not for all HLAs. For most HLAs, we don't have any data. Now there is this growing body of mass spectrometry data, so which I'm calling here eluted ligands, the EL data. And this is probably will become bigger than the first circle there. And these have been used to train these sequence-based prediction methods. But there are other things that we wanted to predict here, for instance, the peptide immunogenicity or the capacity to trigger T cells and the stability of these peptide HLA complexes, because this also affects immunogenicity and so on. So people have developed ML models to predict immunogenicity and stability, but the lack of data, the very limited data sets have, hindered the accuracy of these models. So what we investigated here with Romanos Fasoulis, also CS student, was can we use what we have available on binding affinity and eluted ligands to create a predictor for immunogenicity or stability? So what he did first was like training a machine learning model with the data available from binding affinity and eluted ligands, the same way that was done for previous sequence-based methods. But then he fine-tuned this model using data for immunogenicity or stability, and creating then a new predictor for this kind of transferring what was learned in the previous data set for this new application. And we have very good results with this as well. I will not go into many details because this is unpublished data, but we are outperforming other available tools for immunogenicity prediction and stability. And I'm very interested to see if we can push this further by also adding a structural layer that was not investigated in this particular work. So I'll finish here. Thank my collaborators, especially the Kavraki lab at Rice University, and the funding agencies that made this research possible. So thank you.
Thank you very much. Personally, I want to give it up to all our speakers. It's been wonderful. It's been wonderful, and I've learned a lot. At least I can pinpoint like 2-3 points I personally will incorporate into my research, and I very much believe like everybody wants the speakers are so they've been so wonderful and very fantastic presentations. Thank you very much. Thank you so much. Now it's time for question and answer. We have some questions here already sent to us, and I think we are going to start from there. So after that, we can throw the floor opens to any of our attendees or anybody that also want to ask their questions directly. So I'm going to start with the first questions. First question says, the question is talking about the adaptability of the model. I think that probably is going to Dr. May. I think this is asking about the adaptability of the model and the interpretability application of the models. So that's the way you put it. I don't maybe you understand. So just asking adaptability of the models and interpretability application of models.
So it depends on which models you're talking about. So for instance, the adaptability of the models. So if the question is related to, can we, let's say, let's take, for instance, the host pathogen model. We can adapt it to different hosts, right, with potentially different conditions, depending on what mechanisms you incorporate into your mathematical model. In terms of the interpretability of them, so the models, the mechanistic models are adaptable, depending on what data you're putting in, and potentially they could be robust enough to be used in different environments, different conditions that the host or the pathogen is seeing. In terms of interpretability of the model, I think I didn't have time to go into it. I was probably speaking too fast. But one thing that we were able to do with, let's say, the host pathogen AVM model is actually see the emergence of new features, right? So the host pathogen model, you can actually interpret things in terms of whether you have containment, where you have the formation of a structure that looks like a granuloma, whether you have clearance of the infection. So those types of multi-scale models actually provide a lot of insight into the interactions and what occurs in the disease. We also are able to observe new things that we didn't program into the model, right? These are emergent properties, such as where we saw something that looked like a latent infection or looked like a granuloma forming and then a reactivation type of event. So depending on as you build the models, usually you have a phenotype that you're interested in measuring, and definitely the model would incorporate that phenotype. So so I do think that they're able to be interpreted in terms of things that could be clinically relevant.
Thank you, Mo. Thank you.
Thank you, Dr. May. Yeah, we have another question from one of our participants. He says, I am a synthetic chemist. I often prepare a panel of potential drug candidates for in vitro screening. How can I learn and be involved in computational system biology to facilitate the drug discovery process? Anybody has a take on?
I've pasted the question on the chat so that I can understand properly.
Sure.
Okay, maybe I can put an input here. So look, the thing that we do these days is, especially like I'm from a chemistry department. So we decide to collaborate with our synthetic chemists so that when they're designing certain things, we test it computationally. So usually if you're a student, we try to make a joint project that the student will do both computational and experimental. That way it assists you in knowing both sides of the fields, right? You don't just have one side.
Okay, thank you very much, Dr. Krishna. So the next question is saying, what is your advice for starting in drug discovery field? Probably the person is not in drug discovery field and he or she is interested in that, and he or she is asking, what is your advice in starting in drug discovery field? To any of our panelists and any of our speakers.
So I don't know if I really have really perfect advice for this one, but I guess one approach would probably be try to seek out some kind of internship. There are companies that have internships specifically for people from non-traditional backgrounds, meaning so maybe someone who was not initially targeting drug discovery to begin with, right? So that might be an option that that came off the top of my head. I don't think I really have a good answer for that question.
Thank you very much, Dr. Frank.
Yeah, we have another question that says in the chat, it says, I'm curious, hi Aaron, I'm curious about how the binding pockets in RNAs compare to proteins in terms of size and properties. That's the first question.
Yeah, let me try to start with the first one. So I haven't done this analysis myself, but I know that data is available in the literature, right? And so people have done this kind of analysis. What I have done in the past is actually, take a database of protein ligand complexes and RNA ligand complexes. And what I try to do is fingerprint those individual pockets using the fingerprinting approach that I kind of alluded to in my presentation, and then compare these two, right? And so what I've found is that there is a separation between the protein pockets and the RNA pockets based on this fingerprint. Interestingly, some of the one of the RNAs that I alluded to in the beginning of my talk, where there was this rival switch where they discovered this synthetic small molecule that binds, that particular RNA had a fingerprint that was most similar to proteins, right? So I'm not sure exactly how to interpret that, but there are examples of some RNA pockets that resemble proteins. And this is one of those associated with a rival switch. And it's been shown to be bioactive, meaning that small molecule is actually able to effectively inhibit replication. So the other part is about how do you build specificity? Yeah, so I think, the specificity here would have to come not necessarily from the bases themselves, but the geometry, right? So if you compare different RNA pockets, there is quite a variety of different geometries of those pockets. So that's where the specificity would come from, not necessarily, the specific hydrogen bonding interactions. It's probably a more of a shape-based type of specificity.
Okay, I think you've answered that question. I think the next question is.
Kenny, I just had a follow-up question to that.
Okay.
Yeah. So regarding the first question about the models that you're building for, say, the RNA, and when you looked at, you were able to see that one of them was similar to a protein. Do you build regression models where you can get more information about which properties are more relevant, or do you build, say, something like a neural network model where it's a black box and the model is what it is?
So the general models we build are random forest models, and we use a lot of random forest models. And so, one of the things we did for our classifiers, go back and ask, what are the important features, right? So in our paper, we actually do this importance analysis, and we can identify specific features that are relevant for post-prediction, but also relevant for cavity identification. And so in principle, we can go back and actually figure out what are the differences between RNA pockets and protein pockets, but we haven't done an analysis as yet. But it is possible to do it.
Thank you.
Okay, I think the next question is also to Dr. Frank. The question is, I would also like to know from Dr. Frank, is the number of RNA structures that you currently have compared to proteins sufficient to train the models that you are using for AI?
Yeah, so as I alluded to, right, that's one of the problems that we don't have a lot of data, right? But if you think, for example, for the RNA cavity miner, right, we used about 88 RNA ligand complexes, right, for that study. But for each RNA ligand complex, we have the known pocket plus a bunch of decoys, right? So in total, you have it's 88 times number of pockets on that RNA. So it's smaller than you have for proteins, but it's sufficient to be able to train robust classifiers. Again, there's always it's highly likely that there's a limited domain of applicability of these models, right? Where if you go beyond structures that resemble what you have in your training set, you might expect that your performance retards significantly. But for what we're trying to do, we really want to be able to rapidly make predictions, right? So even if there might be some limited scope for the models, they're still useful.
Okay, thank you. Sorry, Ryan, before I go, I don't want to miss this question. I think I omitted one question. It says, sorry, it says, the number of RNA structures seems a bit low. Was your count only isolated RNA molecules with small molecules bound, or does it include RNP structures such as the ribosome?
Yeah, so that count only included RNA only and RNA small molecules, right? So I didn't include RNA with proteins in that count. So yeah, it's really small, right? It's not a lot of data to work with.
Yeah, so there was another question for Dr. Antunes, but I have just seen that he responded in the chat. There is a question also for Dr. Kim. For your fusion PDB, I was curious if you have taken a look at the predicted alignment error, PAE, for your AlphaFold models. Both AF2 and Rosetta 4 typically struggle with domain packing, which I guess would be relevant for fusion protein. Yeah, something.
Yeah, actually this because we are predicting the novel protein sequence. So I mean, yeah, we also wanted to know how to validate these things. We are getting this kind of question a lot. Anyway, so we use the PLDT scores rather than PAE because I think the PAE requires the known structure, but so far not many fusion protein structures we have. So in our fusion PDB, we wanted to provide systematically for those some number of fusion proteins. So we are just using PLDT. But yeah, I agree. Like for some cases, yeah, we need to check this. Yeah, PLDT. Yeah, I'll try.
Thank you. The next question I think is for Dr. Dinler. And the question is DINC, that's the DINC 2.0, sensitive to which fragment of the peptide you first dock?
Yeah, he responded in the chat.
Yeah, I gave a longer answer in the chat, but in short, it can be, but there are some parameters that you can tweak on the software as well. Like, for instance, the size of the fragment. If you start with a very small fragment, it's more potentially biased by the initial fragment. If you start with a slightly bigger, it's not as much. And then we have different algorithms to choose this fragment, for instance, based on the potential for hydrogen bonding.
Okay, thank you. Then the last question before we throw the floor open to anyone that want to ask questions directly, and this is going to, I think, is going to Dr. Aaron. Will you use the transfer learning to leverage the structural data on proteins to make predictions about RNAs? I guess not for folding, but maybe for druggable binding.
Yeah, that's definitely an option on the table. I've come across some recent work that show that you can indeed transfer some learnings from protein ligand complexes to RNA ligand complexes. It's in preprint, but I'm excited to take a look at that because that's definitely an option, right? Where you say, okay, well, I don't have a lot of data for RNA ligand complexes, but I do have for proteins, so I will transfer what I've learned for the proteins to RNA. So that's definitely an option.
Thank you. Ryan, do you have any question we're missing?
Not in the chat right now, but I have a question, a general question. What is your advice for a medicinal chemist to rationally design an RNA targeting small molecule or a small molecule targeting the fusion protein? What are the main challenges on that?
So I could talk about the one for RNA, right?
Yeah.
So the challenge there would be structure, right? Goes back to the fact that we don't have a lot of data, right? So very often you have an RNA that you think has interesting biology, but you don't know the structure. And so without the structure, you can't deploy the same kind of tools that we've kind of talked about today, like docking and simulations. So that'll be the major challenge. Now, if you have an initial hit, then you can use ligand-based approaches, as I think Krishna kind of alluded to at some point. And that would be the strategy I would take in when I don't have any structure. But very often the issue to that, the challenge in that approach is the fact that you don't have structural information initially to guide you.
Thank you.
I think there is another question for Dr. Pora. And the question says, for your fission PDB, I was curious if you have taken a look at the predicted alignment error for the AlphaFold model. Both EF2 and Rutherford typically struggle with domain packing, which I guess will be relevant for your fission protein. Did you see the question? Or should for you?
Where can I mean, could you say again? I mean, the key question
was-
parts.
Let
Yeah.
me drop it on
Answer
the chat. I've dropped it on the chat so I can see. It says,
Oh
for your
yeah, I
fission-
can see it now.
Okay.
Look at the-
For your fission PDB, I was curious if you have taken a look at the predicted alignment error.
Oh yeah. I think it's the same question that I responded before,
Okay,
right?
sorry.
It's the
Okay.
same question, yeah. So there are not many fusion proteins available now. So we didn't provide the whole systematic, but yeah, I think it's worth it to check the known cases. Yeah, it's the validation. Yeah, thank you.
Okay, thank you very much. Thank you very much. Now I want to throw the floor open. If anybody has questions, can you please signify so they can ask from our speakers? We've attended all the questions sent to us. Do we have anybody that want to ask any question? Any question? We still have probably 12, 13 minutes before we wrap it up.
I have just a quick question to Dr. Kim. I don't know if you talked about it, but do you know how many fusion protein are so far reported in the database structure?
Actually, like because the longest history fusion protein is the PCR-ABL1. Even we searched that, we couldn't get the whole structure. I mean, many of them are like Gleevec combined, the Gleevec and the ARC domain, ARC kinase domain complex things are most often. And yeah, I think not many really, like maybe less than 100 for sure, but it will be smaller than that. So yeah, we have to work a lot. Yeah.
Any additional question? Any question from anyone?
I want to have ask like the person who asked me about the PA thing. So she said like AlphaFold and Rutherford typical struggle with domain packing. So I mean, yeah, usually when I check the PDD score distribution, seems like the domain area has the high score, but not other area have typically low scores prediction. So I mean, yeah, actually, I wanted to directly work with those teams regarding fusion proteins. I think because this is very specific topic. So yeah, I mean, I wondered what's the exact meaning of domain packing thing. Yeah, if
okay, I think we have someone that want to ask question. John Obasumi, right? Can't hear you. Can you speak? Yeah, speak louder, please.
Good evening. Can you hear me?
Yeah, we can hear you clearly.
I was going to ask about, I think there is a question raised the other time concerning the newbies. Some of us that are not really into this drug design and we are interested in joining. So when they were talking about it, then the network was cracking. So I think I need a recap on that.
Okay, I think it was referring to the question about advice for those that are interested in drug discovery. And I think he said his network was poor when the answer was given to that, I think by Dr. Frank. So he was asking if probably you can repeat your response to that.
Yeah. So my suggestion, I don't know if it's a great one, but is trying to seek out some kind of internship. There are some internships that are specifically geared to people who may not have a traditional background, right? Meaning that they may not have had a path that was leading them to drug discovery and they're making a change now, right? And so some of these internships exist. So that would be one of my suggestions.
Thank you very much. Do you have any more question from Any of the attendees?
Can I maybe add to Dr. Frank's thing about getting into drug discovery? So, especially if you're a postgraduate student, as I mentioned earlier, you always have people doing experiment, right? So you just get your supervisor. There has to be some computational person somewhere in your department or in some other department. If they can establish a collaboration, then that's your other way to actually get in there somehow.
Thank you. Any question? I think we don't have any more questions coming up. Thank you very much. Okay.
Wait,
Okay.
I have another question. Yeah, it's just a curious question to people who are familiar with AlphaFold or any predicting model. How do we go from a linear sequence of amino acids to a 3D structure of the food protein, knowing that in the body, they are like the chaperone protein that can assist the folding of the protein. And in the prediction, we don't have that assistance. So how do we overcome that? If anybody has any?
I'll chime in. So my interpretation would be that you can still get it right without the chaperones, because chaperones are a kinetic contributor and not thermodynamics. So as long as you know the energetics of proteins, it doesn't matter how you get there, as long as you get there for predictions. So it might be the right fold, but they could never get there without a chaperone. It doesn't matter. It's still the global minimum or the optimized structure. So that's how I'd expect to rationalize that.
And just to add to that, like exactly like AlphaFold is not predicting the folding path, right? It's predicting just the final 3D structure, and then is learning from the multiple sequence alignment what are the correlations and amino acids should be close to each other. It's a much harder problem to predict the path for folding, and that's would require a different tool.
Thank you.
Thank you.
Sorry,
very
because
much.
AlphaFold also makes use of GROMAX, right? Primarily. So that's an MD engine. That's basically most of it is just MD that it's doing. So there's many other parts that one can use.
But then is mostly just like an energy minimization of the model that was created, right?
Because it's just a force field. So it's just molecular mechanics. There's nothing else happening there.
Any question, any contribution? Before any contribution or question? Yeah, thank you very much. From GCC, we want to say a big thank you to all our speakers, Dr. Krishna, Dr. May, Dr. Whitford, Dr. Frank, Dr. Kim, Dr. Dinler. Thank you very much. We appreciate your coming. We appreciate, I know, like me and many other persons, we've learned a lot from your presentation. Thank you very much. Thank you very much. I don't know if Stan is still on the online. Maybe you have something to say before.
Nope, just want to say again, thank you all for participating. A very good session. So thank you. And Kenny, Ryan, thank you very much for organizing it.
Thank you. It was a pleasure.
Thank you, guys. Bye.
Thank you.
Thank you. Bye.
Bye.